Well, okay, then I will introduce John Trojanowski, who's a friend and a colleague and a neuropathologist and at the University of Pennsylvania. And John has so gratefully agreed to come talk to us today about the different pathologies that we see in all rare dementias, which is what he studies. So I am going to give the floor to John. We'll talk about 40 minutes and then take questions after that. Good morning.
Good morning. I guess one other thing, I've visited your lovely farm and met David, your husband, and it brought back many warm memories for the year when I was 16 years old. My father retired from the military, and I lived on a farm for a year. As a city kid, it was a thrill, and I had my own horse, and I went riding every day bareback and with the saddle too. It was a lot of fun. But now let me get into the lecture that I'm going to give. And Jamie has told me, and you'll correct me if I'm wrong, that many of you are knowledgeable about FTD, neurodegenerative diseases in general, but FTD in particular. And I think Jamie said some of you may have family members who have FTD or may even have FTD yourself. Do I have that correct, Jamie? I'm not hearing you, Jamie. So we have
it's actually PCA, but posterior cortical atrophy, not FTD.
So in the framework of FTD, that would fall into the FTD rubric by many people. Maybe some people will disagree with me, but I'm familiar with PCA, and our folks don't lump that with Alzheimer's or Parkinson's, but with FTD. But PCA is a very specific, as we've discussed, very specific syndromic disorder that is represented by accumulations of tau pathology, but also occasionally TDP43 pathology. And let me just say that the focus of what I'm going to talk about now is to bring to your attention the fact that neurodegenerative diseases are rarely monopathologies. Alzheimer's, of course, is a dyad of pathologies, plaques and tangles, and we think of PCA as tangles, but rarely it could be TDP43. And we think of Parkinson's disease as Lewy bodies and multiple system atrophy as gliocytoplasmic inclusions. But as you'll quickly see on the next slide, there are, and who advances? I advance?
Try you first. That's what you're
okay. Okay, very good. So this is not in the middle. Can you adjust this in some way, Chris, so it's a little bit more?
We can take it out of full screen mode. How about that?
Okay, take it out of full screen mode. Then how do you, I need your help with that. I think out of full, okay, that's
Is that good enough for everybody to read?
No, the other way was better.
Well, I will read, the only text that you need to pay attention to is the highlighted yellow text. I'm not going to bore you with all the details that are in this slide. So if you would allow me, this may be the only one. If you prefer, we'll go back.
Yes, go back, please.
Okay.
Good.
So, okay, this is from a review in 2000 titled Fatal Attractions. It was a riff on the movie Fatal Attractions, in which two people were fatally attracted to each other. And what I was trying to convey here is that in these diseases, neurodegenerative disorders in general, the disease proteins appear to also have fatal attractions for one another. And that is, Alzheimer's disease commonly has Parkinson's pathology, and Parkinson's disease commonly has tau pathology. So it was a riff on a movie title, but the main point is we don't really deal with diseases that have only one type of inclusion. And I will say at the outset that this, to me, may represent one of the largest challenges in finding therapy for neurodegenerative diseases, because most of what you've heard about is A beta focused targeting, for Alzheimer's disease, or tau for Alzheimer's and other tauopathies. But if there are other protein aggregates on board, other pathologies on board, they are, to my view, unlikely to respond tau is unlikely to respond to an A-beta targeting therapy. So I will, go to the next slide, since they're sensitive. And this illustrates, I think, in one slide what I mean. PCA is not here. This is a lecture that I give to a more general audience, but it would be among the frontotemporal degenerations because it is a tauopathy. And you see that Alzheimer's disease actually is a disease that commonly has 4 proteins that aggregate. A-beta and tau are diagnostic, so you have to have A-beta and tau aggregates for the diagnosis of Alzheimer's disease, but alpha-synuclein and TDP-43 are commonly there. Hence the challenge of a therapy that focuses only on A-beta. So in this expert review, if you will, I was able to get together the people that you see as the as the authors here, including people from the FDA, and we made the case that combination therapy has proven to be an effective strategy for treating many of the world's intractable diseases, from TB to HIV/AIDS and so forth. And it's not surprising that the disappointing results in Alzheimer's disease, I'm using that as the prototype because most of the therapy clinical trials that have been done in Alzheimer's disease, don't use combination therapy. And then I cite another paper here talking about recent advances in multi-target anti-Alzheimer's disease compounds. So the beginnings, this is a paper in 2013, the beginnings of efforts to use more than one drug target, that is more than one disease protein as a target for therapy to treat these diseases. This is from a Cummings review in 2017, so it's fairly recent. You don't get a snapshot of what's happened in 2019 until 2020, late in 2020. This was, I think, the December 2017 paper, so capturing some of 17 and everything earlier. And I think it's important to look at the outer circle, which is phase 1. The next circle in is phase 2, and the inner circle is phase 3. And the key for this, the table is on the right-hand side: amyloid-related, tau-related, other targets. And these are the subjects that were studied. You may say, well, why were healthy volunteers included? Because some of them may have had biomarkers indicating the presence of disease pathology, such as amyloid. One can do A-beta amyloid imaging and detect people with amyloid pathology, and it turns out that about 40% of people over 60 have amyloid pathology in their brains. What is unknown is how many of those people will persist with that amyloid pathology and remain normal, and how many will convert to Alzheimer's disease. Prodromal disease refers to people who actually do have mild impairments. So the efforts in the past were to treat mild to moderate disease in clinical trials, and these have all uniformly failed. So the field is moving up the pipeline to try to find that sweet spot where there is pathology and very mild or no impairments, with the hope that the pathology can be eliminated before neuron loss occurs and symptoms occur. This is just a very busy slide, and all you have to appreciate is that there are 117 drugs that are in clinical trial. That's good, but too many are the red ones. The red ones are the amyloids. We don't have enough diversity. There's tau-related too, of course, in the blue, but there's very few blues, and there's lots of others, which means a hodgepodge of different things, and you can see the names of those different things here. But there is not enough tau-focused therapy, there's not enough immune-focused therapy, and our field has to convince the drug companies to open up a wider pipeline for more therapies. The scariest thing about this slide is the outer circle, phase 1. Cummings says that if you're not in phase 1 now, you won't have a readout of your clinical trial until after 2025. I'm giving you an indication of the timeline it takes to go from a clinical trial in 2019 to an outcome, and the outcome would not be known until after 2025. So this is the reality. This is just the reality of clinical trials. It's how long it takes with the current clinical measures we have to see evidence of clinical improvement. You can see a biomarker change much more quickly. Maybe in a year you could see tau reductions or a beta reductions, but so far we have not seen, after 3 years in a clinical trial, statistically significant clinical improvement. So that it creates a need, I would say, for better neuropsychological testing so that we can see earlier evidence of improvement than we're now able to do with memory tests that we do for Alzheimer's disease. And the kinds of things that people are thinking of are people's ability to handle financial matters. So financial dementia may manifest first with people's inability to manage their checkbook or bank account. This is still in development. I'm not saying there's a clinical trial yet that is using tests of financial abilities to develop a readout for a clinical trial. My major point here is that we can see a biomarker change, and that's great, but the FDA will not approve a drug that only eliminates amyloid without improving the cognitive abilities of the patient, and the same is true for tau, TDP-43, or any other neurodegenerative brain pathology. So the FDA's stance on this is no clinical improvement, no approval of the drug, no matter what the biomarkers do. That is the FDA's stance, and you can argue with it in many different ways, but I think what most patients would really want to see is not a biomarker improvement, but a tangible clinical improvement in their impairments. So I am just going to tell you that we are blessed in our center to have all of these different diseases, and I'm not going to list them, but just to tell you that we have a brain bank of about 1,800 brains from people who were parts of studies funded by the NIA, largely by the NIA, but also NINDS, that allowed us to follow patients longitudinally. And many of our patients, in the interest of science, in the interest of helping their own family members, have consented to autopsy at the time of death. And they insist that we make their brain tissue available to others and just not use it ourselves. And we do that to the best of our ability. So we share tissue with other Alzheimer's centers. So I'll just tell you, I direct the Penn Alzheimer's Center. The Penn Alzheimer's Center is really an Alzheimer's disease and related dementia center. That's what we've evolved to. That means that we do follow PCA patients in our center. We follow other FTD patients and also dementia with Lewy body patients. I am pleased to tell you, I don't think I've told Jamie this yet, she certainly knew, I was struggling with a U19 grant, a very large multi-component grant that has clinical and basic projects, and I failed to get it 3 times, and on the 4th try I succeeded. I've been doing this for 40 years. I've never before had to go in a 4th time for a grant. I usually get it on the 1st or 2nd try. This was very stressful. We spent $2 million of philanthropy money to keep our program going. That's what it took to keep the program going, and I'm pleased to say we have completed the paperwork to get our funding probably in the middle of September. It is an $18 million 5-year grant. Virginia Lee has an FTD grant that would have PCA in it. This grant is on synucleinopathies, which would mean certainly Parkinson's disease, but I've perhaps not mentioned this yet, but Alzheimer's disease is the most common synucleinopathy because there are 5 million Alzheimer patients and 50% have Lewy bodies. So Alzheimer's disease is a more common cause of Parkinson's disease or synuclein pathology than Parkinson's disease itself. There are 1 million Parkinson patients. The number of patients with FTD is far less, perhaps 100,000. Our center, as well as Bruce Miller Center and Mayo, are a couple of the centers that really have outstanding strengths in FTD, including PCA. So we have all of these tissues to make use of to do many different types of studies, and the study that I'm talking about now, of course, is the one where I illustrate the co-occurrence of neurodegenerative disease pathologies in different neurodegenerative diseases. Except for this slide, this is a comparative survey of the topographical distribution of the signature molecular pathology in major neurodegenerative diseases, and you'll see those diseases shortly. So these are patients that we studied who, if they had Alzheimer's disease, just had plaques and tangles, who, if they had FTD, just had tau pathology or TDP43 pathology. I know you're all interested in PCA. Jamie knows that I am not a clinician. I'm an MD, but my emphasis is on pathology studies and animal model studies. So I cannot wax poetic about PCA clinical profiles. I am sorry for that, but we all have areas that we specialize in and try to excel in, and mine is in the pathology of these diseases, which do include, of course, PCA. So the panel of slides on the left shows Alzheimer's disease pathology. This is a thioflavin stain, an amyloid stain, and thioflavin will stain all amyloids, not just A beta, but tau and synuclein. These are all of these inclusions form fibrils that render them, by definition, amyloids. This is A beta amyloid stain with an antibody to A beta that we produce. This is one stain with an antibody produced by Peter Davies. And what you see in the bar graphs, and maybe we shouldn't fuss with them too long, is the differential abundance of these pathologies, the A beta pathology, the tau pathology. The brain maps are perhaps the easiest way to see the differential distribution of these pathologies in the cortex, a lateral view, a medial view, a coronal view, and in the brainstem. And then the same is true on the right-hand side, where PDD, that's Parkinson's disease with dementia, dementia with Lewy bodies, and MSA are illustrated. So I'm giving you an idea of the heterogeneity of the distribution of these Lewy body pathologies on the right and the plaque and tangle pathologies on the left in these different diseases. And then here is Pick's disease, CBD, PSP, and FTLD, TDP, and ALS. So I'm sorry, I don't have a slide of the disease of most interest to you. We do study it. We have published on it. Jamie knows that. I just don't have a slide in this slide set on the distribution of that pathology, posterior cortical atrophy. But it's very simple. It would mean that most of the pathology is in the posterior cortex, hence leading to the atrophy that gives the syndrome its name. So this is a busy slide too, and I think I can step you through it to make it easy. This is neurodegenerative diseases, concomitant proteinopathies are prevalent, age-related, and apolipoprotein E4 associated. So the take-home message is pathological tau, A-beta, alpha-synuclein, TDP43 was studied in 766 longitudinally followed patients with diverse neurodegenerative diseases, or ND, that we acquired from 2000 to 2013, and then partially blocked in the slide is the number with Alzheimer's disease, Pick's disease, and so forth, just to give the details of the diversity of the brain bank that was interrogated. And the bottom line is that these people had the pathology that made the diagnosis of Alzheimer's and MSA and so forth justified. But our data also implies that increased age and ApoE4 status are risk factors for the co-pathology that we see independent of the neurodegenerative disease pathology itself, and that the severity of the neurodegenerative disease pathology influences the co-occurrence of other pathologies. So the more synuclein pathology a Parkinson patient has, the more likely it will be that they have co-pathology, and that co-pathology will be severe. I can say a word or two about ApoE if you're not familiar with it. It is a risk factor allele for Alzheimer's disease, initially described as a risk factor allele for Alzheimer's disease, and then was shown to be a risk factor for Parkinson's disease. It's also a risk factor for heart disease. And so there are, as everyone knows, there are 2 chromosomes for every gene. So there's 2 chromosomes for ApoE. There's ApoE2, 3, and 4. They differ by 2 amino acids each. And the E4 allele, 1 copy of that increases your risk for Alzheimer's disease by 10%, and 2 copies increases it by 20%. So this is one of the most serious risk factors for Alzheimer's disease there is. The worst gene change to have, of course, is a autosomal dominant mutation, such as an A-beta or PS1, that would mean that you absolutely get the disease by the time you reach 40 or 50, no escapees. So this is autosomal dominant disease. If you have the gene with the mutation, you will get the disease, period. And this is being exploited in Colombia, in Medellin, Colombia. There are about 7,000 individuals from a very large Spanish cohort that migrated to Medellin, and many have died, of course. They have a mutation, I didn't say it's family, with a Alzheimer autosomal dominant mutation. I think it's PS1, forgive me for not knowing the specific mutation. And many patients have died. The typical scenario is they get memory impairments, mild memory impairments by 40. By 45, they are demented, and by 50, they are dead. By 50, 52, they are dead. So this group of individuals includes about 800 who have the mutation but are too young to have clinical manifestations. So they're people under 40 years old. And these people are part of a clinical trial that we call a prevention trial. And we would call it a secondary prevention trial because we have a biomarker to indicate they have disease. So we know by the fact that they have the mutation that they will, by age 40, develop mild memory impairment and progress, as I mentioned earlier. So these people are in a clinical trial, and the drugs that are being tested have been A-beta drugs, A-beta targeting drugs, so anti-A-beta antibodies or BACE inhibitors. I think they now are adding tau, anti-tau therapies to this group. Oops. Well, let me skip to this next slide because this, I think, makes the case easiest. These are the diagrams of the distribution and abundance, not the distribution, the abundance of A beta pathology, alpha-synuclein and A beta pathology, and pure TDP-43 pathology in a disease called amyotrophic lateral sclerosis, which is diagnosed by the presence of TDP-43 pathology. But when you do a bar chart like this, you find they all have TDP-43 pathology, but the vast majority also have other pathologies. And that's repeated in the subsequent slides here, FTLD, TDP, and Lewy body disorders down below. And this is the key for these pie charts indicating the frequent co-occurrence of other pathologies in these disorders. And these are other examples of other neurodegenerative diseases with co-pathologies. This is people with very mild pathology, so they don't have very much at all. This is intermediate Alzheimer's disease, high Alzheimer's disease. Pick's disease is almost pure tau, but there is A beta in these. They all have tau pathology, but 25% will have A beta pathology. Oops. And this is a graphic to illustrate the common presence of Alzheimer's disease neuropathological change in the diseases that I've mentioned previously. They are shown down below here, and you can see there's a variable burden of this pathology. Red is high, orange is intermediate, yellow is low, and this refers to very low tau pathology in a disorder we call PARC. So each of these diseases has associated co-pathologies, perhaps CBD and PID, Pick's disease and cortical basal degeneration have the least, whereas Lewy body disease, Parkinson's disease has quite a bit of mixed Alzheimer's disease pathology, and as you might expect, in Alzheimer's disease, the abundance of Alzheimer pathology is high, and even in intermediate Alzheimer's disease, it is high. No surprise there. We did a study with Claudia Kawas at Irvine to ask what is different about these pathologies and the manifestations of Alzheimer's disease and what are called the oldest old, that is people who are over 90. It would seem to the field, there aren't lots of people over 90 that have been studied and been studied for a long time, but it seems as though we know, well, it doesn't seem, we know that the prevalence of Alzheimer's disease goes up after age 60, and it seems to go up exponentially until about age 80, when 40 to 50% of people have Alzheimer's disease. And I just don't have those numbers for PCA. They just, I doubt if there is a study like this, like the one I just mentioned for Alzheimer's disease for PCA. I may be wrong, but Alzheimer's has been the most studied in terms of aging-related accumulation of Alzheimer pathology and clinical impairments. And so in the oldest old, these people are complicated not only by the fact they have Alzheimer's disease, but they have increasing amounts of other pathologies like cerebrovascular disease, hippocampal sclerosis. The hippocampus is the memory machine of the brain, sitting just in front of your ear on either side, in your brain, of course. And sclerosis means scarring, so they have scarring of the hippocampus, which means they've gotten scarring because neurons have died, resulting in gliosis. And then there is another entity called RTAG, or aging-related astrogliopathy. It's not been very well studied, although I'm proud to say we have studied it. And in this particular study I'm referring to, we found that RTAG did have a clinical impact. There was an added burden of clinical impairments that was associated with RTAG. These patients also had TDP43 and LB, Lewy bodies. So all of us would love to live to be 90, at least I would love to be 90, like to live to be 90 and active as I am now. I don't know how well you can see me, but I am 72 years old. The grant that I just got will be renewed in 5 years when I'm 77, and I fully intend to renew that grant, which would take me into my 80s. The NIA does not discriminate against people based on age, and I have been told by the NIA program director to go for it, John. When I get next year, I'll renew my Alzheimer's Center, and the program director said, go for it, John. So there is not age discrimination as long as I'm competent and able to do my work. And it would be very evident when I can't do my work. My colleagues would certainly make known to me that I'm failing, and I would know I'm failing. And I have made a promise to myself that if I can't do the job, I'll just stop what I'm doing, the work that I'm doing in neurodegenerative disease. So to make a long story short, we found, as I said, these many different pathologies in the oldest old, and it modified the status of their Alzheimer pathology, mostly by increasing the burden of Alzheimer's disease. And that is what is stated at the bottom. Our findings imply that non-AD pathologies, including cerebrovascular disease, contribute to cognitive resilience in the oldest old. That is, the absence or presence of that can make the Alzheimer symptoms better or worse. I include here a slide on RTAG because most of my colleagues are unfamiliar with what RTAG looks like. I showed you earlier a plaque, a tangle, a Lewy body, a TDP43 inclusion, an FTD tangle. And so what I'm showing you here is the RTAG pathology that is seen in astrocytes. And as I've mentioned, it now is associated, at least in the 90+ group, with more severe cognitive impairment. So it's another player to be considered when one wants to think of therapies for neurodegenerative diseases. And there's a little review down below here. If you use PubMed et al., that is the way to access the scientific literature. You can search for me or Gabor Kovacs, who was the person who did the most early on to identify RTAG, and we collaborated with him. This graphic shows that the stage of Alzheimer's tau pathology, Braak staging, has been used for Alzheimer's disease. I collaborated with Braak in an FTD staging paper so that we could stage FTD pathology, tauopathy pathology. And this is important for ascertaining in a quantitative way the burden of Alzheimer pathology. So obviously stage 1, 2 is far less pathology than stage 5, 6. And what these lines are showing is that as you increase in stage, that is the amount and burden, the frequency of tau pathology, the amount of co-pathology, the scores for co-pathology increase. And that is seen here for cerebrovascular disease, for example, here, and Lewy body pathology here. And we're showing hippocampal sclerosis here in the bar graph by these dashed lines. So non-AD associations with dementia in both the not AD and dementia groups are shown here. I think the point of this slide for me was to indicate that cortical archag indeed has statistically significant contributions to the clinical abnormalities in the 90+ group. So I also work a lot on Alzheimer's and Parkinson's disease. It's 11:35. If you will allow me to talk another 10 minutes, I will talk to you about the interactions of Alzheimer's disease and Parkinson's disease. I just have not done these studies in PCA, so I can't. We do study PCA, but we don't have a large enough cohort to do the kind of study I'm talking about here. I think we had 140 autopsy confirmed patients with Parkinson's disease or Parkinson's disease dementia at the University of Pennsylvania. We're not restricted to the University of Pennsylvania. You'll see in subsequent slides that we branched out and collaborated with other centers to expand our N. And what we are basically showing are the neuropathological substrates of Parkinson's disease with dementia. And the clinical, pathological, and genetic variables we examined are listed here. And these are our scoring systems. They are subjective scoring systems. They are the ones that are recommended for all Alzheimer's centers for neurodegenerative diseases, not just Alzheimer's, but FTD and Lewy body disease. We do have digital microscopy and computer systems to measure the burden of pathology so that we have absolute quantitation. We do that. I'm not showing any of that in the slides that I'm using in this talk. So I think the takeaway message here is that people with Parkinson's disease and Alzheimer's pathology versus Parkinson's disease without Alzheimer's pathology have a difference in the dementia to death interval. It's much shorter. And the age of motor onset is later with Alzheimer's disease as opposed to just Parkinson's disease. I'm not going to go into more on this slide. I guess I could mention that death, strangely, the age of death is later when people have Parkinson's disease and Alzheimer's disease as opposed to Parkinson's disease alone. These are findings, and other people have had similar findings, and obviously more has to be done to understand what they mean. This is now a multi-center, so I just want to reassure this audience that we are not restricted to just doing studies with our own material. This is the Perelman School of Medicine here. This is Washington University in Seattle, Oregon Health Sciences University in Oregon, the University of Kentucky, and the University of Pittsburgh. These are all centers of excellence. The authors of the paper are down below here. And we were able to look at a larger number of patients here with PDD, Parkinson's disease dementia, and dementia with Lewy bodies. And the take-home message, I think, is readily seen here. If you have a high level of Alzheimer's disease, your survival is shorter as opposed to no Alzheimer's disease. And on the left, what I'm showing is no Alzheimer's disease. The motor impairment onset to dementia level is longer with no Alzheimer's disease pathology than if there's a high level of Alzheimer's disease. So it makes a difference. It makes a difference if you have Alzheimer's, if you have Parkinson's disease pathology alone or Parkinson's plus Alzheimer's disease. These scores indicate that cortical, synuclein, tau, and A beta are correlated. So their abundance, their frequency correlates, suggesting that they may interact. so in Lewy body disease, the full spectrum of clinically manifest synucleinopathies shows that cortical synuclein pathology is associated with dementia, and that AD neuropathology is common. And I can't read everything here because it's partially blocked, but it correlates with a higher burden of synuclein pathology, earlier onset of dementia in a dose-dependent manner. And we still need additional studies to understand what I've just summarized as the conclusions of this study. Tau copathology associates with higher synuclein pathology. The distribution is altered from the distribution of pure Alzheimer's disease without synuclein pathology, and what that signifies to me is that these pathologies interact. They're not just sitting in the brain independently, but somehow interact with one another. And AD copathology may have specific clinical features based on temporally mediated cognitive functions. There is a lot to talk about with respect to genetics. I've already mentioned ApoE. Glucocerebrosidase is a gene that causes Gaucher's disease, as you may know, but it also, there are particular mutations that are associated with motor and frontal executive impairments. SNCA is the abbreviation for the synuclein gene, and there are variants in that gene that are associated with tremor-dominant Parkinson's disease, which has a more benign prognosis than postural gait instability disorder, Parkinson's disease, which is associated with the LRRK2 gene. So there's still a lot of work to do to understand the genetics, how GBA, LRRK2 contribute to the disease, as well as to develop algorithms that would help us use these variants to predict who will progress, for example, slowly or quickly, who will respond to a drug when we have one that is disease modifying, and who will not. Those are the aspirations for my genetic colleagues. They see genetic studies as an arm of drug discovery. I'm getting to the end of my slide set. It's 12:43 by my clock, and this is a study that was published in 2003. This may not mean all that much to you, but what I'm showing you are individual filaments that we assembled using soluble tau or synuclein. And what we were able to show is that the protein tau, that tau fibrils could induce synuclein fibrils, could induce a monomeric synuclein to fibrilize, and the other way around. This is what I mean when I inferred in the previous slide that there could be interactions. This is exactly the kind of interaction that I mean. A tau fibril can induce monomeric soluble synuclein to fibrilize, and the other way around. And these different sized beads, the small and the larger ones, are showing that these filaments contain both alpha-synuclein and tau pathology. And I believe I'm getting to the end of my talk. This is the Contursi family that we've studied, showing, for example, that they have abundant, so the Contursi family has Parkinson's disease. They have a mutation in the alpha-synuclein gene, which is this A53-2T, alanine to tyrosine change at position 53, and this causes an aggressive early onset form of Parkinson's disease that is not just characterized by synuclein pathology, but also by tau pathology. And the best example of that is shown, I think, in panel Q, in which the red are the Lewy bodies, and the green is the accumulation of tau pathology. the interesting thing is they're in the same cell, but they don't commingle, although I showed they commingle in the filaments themselves. I think the last slide is the team. All of this work was supported by grants from the NIH, particularly NIA and NINDS, also the WARE Alzheimer program, the donor who supported our research. And I always say that these studies could never, ever have been done without the support of the families and our patients who urge us on daily when I talk to them to move as quickly as we can. And I will tell you, at age 72, I am at risk for Alzheimer's disease, and my risk grows every day. I said the risk goes up, it goes up, I forget what the word was that I used, but it goes up with a greater degree of frequency from age 60 to 80. So I'm not yet at the highest, I'm not yet at the age of greatest risk for Alzheimer's disease, but I'm hoping very dearly to escape it. I'll just say that there is no therapy for Alzheimer's disease or for PCA. Many of us believe that neurodegeneration can be mitigated, ameliorated, perhaps even prevented to some extent by healthy lifestyles, which means a Mediterranean diet, for example, fruits and vegetables in abundance, meat, of course, fish, by exercise, by doing cognitively stimulating activity. And I'm told that football games are not cognitively stimulating. I don't know that for a fact. The kind of cognitively stimulating activity we think of is reading books, reading newspapers, doing crossword puzzles. You don't have to spend millions of dollars on brain games to have cognitively stimulating activity. You can do it very cheaply for no cost at all, except perhaps for buying the New York Times, which is what I read, or whatever newspaper you read. Also, efforts to reduce hypertension if you have it, to have heart disease treated if you have heart disease, avoiding obesity, and having your diabetes under control. Those are examples of some medically treated conditions as well as lifestyles that associated with a lower risk of developing a neurodegenerative disease, and Alzheimer's in particular. And that is my last slide. And I'm just 2 minutes over. Jamie, I hope that was okay.
Thank you so much. And I just, you can unshare your screen right now so that we can do a question and answer.
Yes.
And
I'm going to put on the light, and you tell me if that makes things better or
no,
not.
Worse.
Worse. Okay.
Yeah.
Light goes off.
It was
like
fine. Yeah, that's perfect. So let me just explain, because I see some people here. Most people understand that PCA, by now you should know that the pathology, most of the cases of the pathology that we've seen, it's a very rare disease. It's not studied much. But the two proteins that seem to come up over and over again are proteins that John Trojanowski talked about, and amyloid and tau. And these two proteins are accumulating in the posterior regions of the brain, which is why your visual and spatial symptoms are occurring. And then it does spread upwards to more frontal areas of the brain. But what John was speaking about today, and which we now understand also, is that there are pathology, there are other protein pathologies like Lewy bodies, alpha-synuclein, that are also accumulating in the brain. And I know a couple people here have had some psychosis, and that could be a matter of Lewy bodies, but we just don't know enough about this right now. I'm just going to go plug my phone in. We just don't know enough about it. So that is why research is important, and I hope that and Bonnie, who's our neuropsychologist at Harvard, and I are always talking about ways to study this condition. So And John has given me a lot of good ideas, so I'll come back to you with that. Right now, what I'd like to do is, if you have a question, you raise your hand and I can see you. I will, yes, Marilyn, I'm going to unmute you.
Okay, hi everybody. So my husband passed away April 30th, 8 years after his diagnosis. He was older than most. He would be 80 in just another month. Very healthy from the neck down, as we say, no other remarkable health. And we had an amazing 8 years. I did just get back the brain autopsy pathology report. We are part of the study at UCSD with Doug Velasco. And
I know Doug Velasco very well. You're in good hands.
I know we are, and he's become a wonderful friend. In fact, Doug is reviewing the study now. I've gone through the 3 pages with Google at hand, so I've learned more about parts of the brain than I ever thought I needed to know. But I thought that everyone on this call would be interested in the bottom line findings, that Lou was diagnosed with a very severe condition of Alzheimer's disease, substantiated by the scores in Thal, Brock, Sarad, and the NIA-AA high ADNC. And I correlated that with a table that I found online. And he's right in that bottom row, bottom right corner. His amyloid angiopathy was severe, and he definitely had the presence of Lewy body co-pathologies. So I do have a case number for that. It will be entered in the NIA database. But if you'd be interested in that case number, I'm happy to read it off to you.
Go ahead.
Okay, that case number is X is in X-ray, D is in David, 5877. And the pathologist at UCSD who did the report is Ann Hineker.
Okay.
And Ann came to UCSD not too long ago from UCSF. Looks like she's at the earlier stages of her career. So I welcome sharing that with you for the benefit of the good that they provide.
Thank you, Marilyn.
We have done studies of autopsy cases in the NAC archives. One study that I didn't mention is very, very important, and that was a study John Toledo led, showing that cerebral vascular disease pathology in this cohort, which includes Alzheimer patients, of course, but also Parkinson's and FTD patients, and even some with ALS, that vascular disease pathology was most abundant in people with Alzheimer's disease, again suggesting some sort of interaction between blood vessels in the brain, heart disease, and Alzheimer's disease. And so that has prompted some people to undertake clinical trials to vigorously treat heart disease as a way to perhaps slow down the dementia of Alzheimer's disease.
Great. Okay. Any other questions? Thank you so much, Marilyn. Connie, you must have a question. You always have good thoughts. Come on.
No, I was just looking. It was actually very helpful. Thank you for you to.
We need to put the sound up a little bit. I can't hear.
Oh, let's see.
Can you get closer? We're at 100% of volume.
Yeah, that's
better. Yeah, go ahead.
Okay. So, no, I just wanted to thank you for offering the information about what things you can do, because people kind of get numb with all the stuff that we can't do anything about. So that was very helpful to include the exercise and the diet and the Mediterranean diet and so forth.
I'm maniacal about it. I'm maniacal.
Good. Well, if
I would like to say that there are clinical trials ongoing right now, not in Alzheimer patients, but in normal elderly, to see if these lifestyles that I've just mentioned, if followed, will reduce the rate of normal cognitive decline. We all have a cognitive decline associated with aging that isn't dementia, but just is a cognitive a loss. I mean, we all experience name finding problems and so forth as we age. And
Right.
so there is a serious interest on the part of the National Institutes of Health, and particularly NIA, in testing objectively whether these lifestyles will benefit, mitigate the normal aging-related memory losses which we have, and then maybe they would be tried in Alzheimer's disease.
Right. Thank you.
Wonderful. Thank you so much, Connie. Anybody else have a question for Dr. Trojanowski? Speak now or forever. Yes, Norm.
Can you hear me?
Yes.
Okay. Well, thank you for doing this. And two questions. One is, are you going to post this online? That was a lot for me to
Yes.
absorb. And I want to see it again. And
I will, Jamie, did I send you the slides? I'll be glad to send you the slides if I haven't already.
Well, we actually have, I'm taping this, so it will be on.
Okay.
Go ahead, Norm.
and my question is about measuring progress or lack of it in the neurologic test. That is so hard to do. And they come out different from time to time, because I think we all have ups and downs. And I wish there were a better way, but besides doing imaging studies every month or something. Do you have any other way or any ideas on that?
Well, so the mania, if I can use that word, among pharmas, at least for Alzheimer's disease, is to emphasize A-beta amyloid. And I have been one of the most outspoken critics of that program since the amyloid cascade hypothesis was introduced in 1999. I said, we can't explain Alzheimer's just on A-beta. Tau is also there, and it is toxic, and there are these other pathologies that I've mentioned as well. So that Jeff Cummings article that I referred to shows that there are other targets that are being sought by pharmas in clinical trials. I will tell you a very uplifting story of the success in the disease that seems so impossible to treat, and that is spinal muscular atrophy. It is an ALS-like condition in children that uniformly killed them by age 4 or 5, maybe they lived to be 6. And it was caused by a mutation in the spinal muscular atrophy gene. And a company called Ionis, I don't own stock in Ionis, I don't know that I own stock in Ionis, I don't invest in any stocks myself personally. I have a TIA CREF account, and there may be stocks, but I have no financial ties. This company, Ionis, has created antisense oligonucleotides. So what are they? They are stretches of nucleic acids that are small enough to enter the brain and shut down the synthesis of genes, or the expression of genes. And so Ionis successfully shut down the production of the mutant form of SMN, and they have treated 30 or 40 patients, and those patients are still alive when they should have been dead by now. So you can believe that this is something that has grabbed the attention of the Alzheimer community and other neurodegenerative disease communities. The problem, so I just said that SMN is due to the expression of a mutant protein. In most Alzheimer's disease, there are no mutations that cause the disease. It's the normal protein that's either overexpressed or undercleared or aggregates and accumulates. And so the challenge will be how much to reduce the expression of tau and A-beta to not cause any toxicity, but to reduce the amount of pathology that would otherwise accumulate with age, if that's possible. So this is a brand new direction for therapy that has cured one really very, very lethal neurodegenerative disease. And we're all hoping to see, and this is happening in laboratories with animal models. I have a colleague, Dr. Miller, who's doing this for tau models at WashU, and I'm sure there is a lot more activity. We're not doing this ourselves. We have a drug discovery program, but it's small molecule drugs that target tau. That there's only so many things one can do in a private laboratory, or even in partnerships with pharmas, which we do partner with. And we're going to be at Ionis Virginia Lee and I just want to say that you saw the names of the co-authors. Virginia Lee is my wife of 44 years and my lab partner of about 38 years. We took us 6 years to figure out we could really be a great team together in the laboratory, and I think we are. And we are going out to see the people at Ionis in September, and I hope we'll open up a line of collaboration with them that would include the kinds of therapies that I've just mentioned.
Awesome. Thank you. Marilyn, did you have another question?
No, nothing at this time.
Okay.
I had a question, Jamie, if I
Go ahead,
may.
Kathy.
Sonia. Hi. So I'm Sonia Ancoli Israel. I've been studying Alzheimer's and sleep for 40 years. But my question actually doesn't relate to sleep. It's in Alzheimer's. It's really a PCA question. I have spoken with Doug Velasco, who's also my husband's physician and a colleague of mine, with Brad Beauvais, who I'm sure you know at Mayo,
Yes.
with Jeff Cummings. They're all
Yes.
colleagues and friends of mine.
Yeah.
And I encourage them to think about doing a study specifically in PCA. Our PCA patients are different, and they're excluded from all clinical trials because of the visual component of the PCA. And so I think there are enough patients now, certainly around the country, certainly around the world. I'd like to encourage you and all our other colleagues in this field to think about doing some clinical trials and studies specifically in PCA. I even offered to write the IRB for you guys. I have a conflict of interest, so I can't be involved in it. But I just wanted to have you think about whether that might be a path for all of you to go down in some large collaborative study.
Well, I think that all clinical trials require funding. And so the beginning of this would be to convince a pharma to commit the funding to do this. And I'm skeptical that they would for the reasons that you mentioned. Mm-hmm. But the NIA will support clinical trials of orphan diseases. So PCA involves few enough people that it would be an orphan disease. I guess I should say that oftentimes a company will engage in an orphan disease clinical trial because if that proves effective, then maybe it would work in Alzheimer's disease. So all of this is on the table and something that you should discuss. I am an advocate, and I've for 25 years been advocating for more funding for Alzheimer's disease and Parkinson's disease. And it's not just me, I want to emphasize, but we've gone from $500,000 in 2000 for Alzheimer's disease to $4 billion now. That's not just me, it's the community of those affected by the disease doing their best to convince members of government and the NIH to commit more funding to what I would say is an epidemic that threatens the health of our nation, I think the security of our nation, and that really should command more resources to shut this disease down as quickly as possible.
Great. Kathy, can you unmute yourself? Because we can't unmute you.
I'm here.
Jen.
I want to thank you for all of the information that you post. I want to thank the people that ask about the drugs, because I think that is one of the main things that most of the caregivers are looking to, is the best drugs for anxiety, the best drugs for memory retention. And I must say that finally it looks like, after being sort of dismissed by my husband's neurologist, he was diagnosed 1.5 years ago. He still has his visual and spatial faculties, but on October 8th, the University of Washington Wellness and Brain Center is having a seminar specifically for PCA. I mean, finally, people are addressing this, and it's going to be, this featured speaker is Dr. Charlie Burnick. And
I know it, yes, very good.
So I just wanted to put that out there for everybody, and I would love to see everybody from the Seattle, Washington area. It's going to be in Linwood, so that we could come together maybe as caregivers for a little bit more support. Jamie, you do just in the PCA web is just wonderful, but the zonal, area of Seattle, I thought was very out nowhere, and then all of a sudden now we have this brain wellness center at the University of Washington conducting this seminar, and I'm just so thankful, and hopefully people will be able to access it. Thank you.
Please take encouragement from all the progress that's happening in Alzheimer's disease when it's not PCA, because if we can cure the plaques and tangles in other types of Alzheimer patients, I would hope that would apply to people with PCA too.
Thank you.
Well, I think that we're going to wrap up if there's any other questions. Thank you so much, John Trojanowski and your crew. And thank you all. And I will post our support group is this Friday. And Brian, if you'd like to join us, anyone else would like to join us, I will just email me at jtalan, Talan 3K at AOL.com. And I will send you direct information on this. Thank you.
Thank you, Jamie. Let me thank you, Jamie. We've known each other for many years. I haven't known this side of you. We've discussed articles, by many different scientists in many different areas. I only learned about this activity of yours when I was out on your farm, and I'm just very impressed with what you're doing. And all of the people on this call are so fortunate to have someone so well-informed to break down all the complicated science and make it intelligible to the non-scientists. You are all very lucky, and Jamie, you are the sweetest and the best.
Thank you, guys. Thank you, everybody. Have a great day.
Bye-bye.
Bye.